to study. Today I will talk about chromothripsis. So can cancer occur in one day? Yes and no. So chromothripsis is chromosome shattering. One chromosome breaks into tens or hundreds of pieces. The cell tries to repair the damage. The pieces are joined together in the wrong order. And this can create many mutations at once. So normal cancer evolution, mutation 1 takes years, mutation 2 takes years, and similarly. Cancer develops gradually. But in chromothripsis, one catastrophic event occurs and hundreds of DNA breaks happen at once. So the cell has to repair everything rapidly and the repaired chromosome becomes abnormal. So the possible causes are: 1. Micronucleus formation. The chromosome gets trapped outside the main nucleus and becomes damaged. 2. Mitotic errors. So mistakes can occur when chromosomes are being separated during cell division. 3. Telomere crisis. The ends of a chromosome are called telomeres. So these become too short and the chromosome can stick together. 4. DNA repair failure. So the repair machinery makes mistakes while fixing DNA breaks. And 5. Radiation or severe DNA damage. So strong DNA damage causes multiple breaks simultaneously. But can chromosomes be repaired correctly? Yes, they can. And most DNA damage is repaired correctly. Cells repair thousands of DNA errors every day. So usually the repair system works well. But then why are they repaired incorrectly sometimes? Because in chromothripsis, too many breaks occur at the same time. DNA fragments become mixed together. Repair proteins become overwhelmed. And the cell repairs quickly rather than accurately. And the cell cycle checkpoints may also fail. So it's like if you rip off one page from a book and have to repair it, it's easy. But if 100 pages of that book are ripped off simultaneously, it's difficult to repair. So what happens after incorrect repair? The tumor suppressor genes may be lost, the oncogenes may be amplified, chromosomes are rearranged, and the cell may gain growth advantages. So therefore, these cells can develop cancers. And which cancers show chromothripsis? Glioblastoma, osteosarcoma, neuroblastoma. Does this mean that cancer can occur in one day? Yes and no. So yes, because the chromosome shattering event can happen during a single cell division. But no, because that abnormal cell that is created still has to survive. So it still has to multiply, it still has to escape the immune system, and the tumor takes months or years to become detectable. So this is important because it shows that some cancers may evolve suddenly, and it challenges the idea that cancer always develops slowly. It supports the concept of genomic earthquakes. So today I'll talk about an interesting phenomenon called Kataegis. Kataegis. So I spoke about chromothripsis yesterday and it's kind of similar to it. Kataegis is like a mutation storm. So imagine a very long book. This book has your DNA written inside it. And normally typing mistakes can occur. So there's one mistake on page 10, one mistake on page 500. These are small mistakes. Mistakes in one letter, let's say, called point mutations. This is how mutations usually would happen. But in Kataegis, imagine instead of a typewriter, the typewriter's 2-year-old child wrote that day. So that particular page has so many mutations. So there are 50 mistakes on one particular page. This is Kataegis. Not a few mutations everywhere, but a mutation storm in one small region. Now why does this happen? So our cells have proteins called ApoBEC. Their normal job is to destroy viruses. That means that they damage viral DNA. But sometimes they get confused and instead of attacking the virus, they attack the cell's own DNA. So in Kataegis, hundreds of mutations appear close together because many mutations occur in a very small region of DNA. So instead of mutations being spread throughout the genome, they occur in clusters. So chromothripsis is chromosome shattering and Kataegis is mutation clusters. These two sometimes occur together. And why is this important? Because this indicates genomic instability. It's not stable anymore. And this instability can create mutations that can drive cancer. So this helps understand tumor evolution and gives clues about mutational processes, how mutations occur. And ApoBC family enzymes are the major cause in many cancers. So Kataegis is a localized mutation storm in which many mutations occur together in a small DNA region. Today I'll talk about genome doubling. So first let's talk about what is normal. In a normal cell, there are 46 chromosomes, 26 from the mother and 26 from the father. So a normal cell will have a complete set of these instruction books, instruction manual called DNA. But what happens during genome doubling? So the cell will copy all of these 46 chromosomes, but instead of dividing into two cells, it fails to separate. So now one cell has 92 chromosomes. Why does this happen? Basically, the cell tries to divide, but the cell division part, the last step, fails. So the chromosomes remain in one cell and the cell survives instead of dying. But why is this dangerous? Now that cell has extra copies of everything. So if one copy is damaged, another copy still works. And this allows the cell to tolerate mutations, survive DNA damage, and evolve more easily. So what happens next? After genome doubling, chromosomes are often lost, some chromosomes are rearranged, new mutations can accumulate, and the cell becomes genetically unstable. And now cancers like this because extra chromosome copies give the cancer more genetic material, greater flexibility, faster evolution, and resistance to therapy. So which cancers show genomic doubling? Some examples are breast cancer, colorectal cancer, ovarian cancer, lung cancer, and glioblastoma. So genome doubling is associated with aggressive tumors and chromosomal instability, and it's linked to poor prognosis, poor treatment outcomes, and it helps tumors evolve rapidly. So in one simple line, genome doubling happens when a cell accidentally keeps two complete sets of chromosomes, giving cancer extra copies that help them survive and evolve. Today I'll talk about another interesting phenomenon called chromoplexy. So imagine 5 different books. Book 1 is chromosome 1, book 2 is chromosome 2, book 3 is chromosome 3, and so on. Normally each book stays intact, but one day all 5 books fall into a paper shredder. Pages from all the books are torn and pieces become mixed together, and someone quickly tapes the pieces back, but mistakes occur. So a page from book 1 goes into book 3, a page from book 4 goes into book 2, etc. Now every book is scrambled. So in a cancer cell, several chromosomes break at nearly the same time. DNA repair machinery tries to fix them. Pieces from different chromosomes become joined incorrectly, and multiple DNA rearrangements occur together. Chromo means chromosome and plexy means weaving or braiding. So several chromosomes become tangled together like crossed wires. Now what can happen after chromoplexy happens? So 1, tumor suppressor genes may be broken. 2, oncogenes may become activated. 3, new gene fusions can form. And 4, the cell gains growth advantages. How is this different from chromothripsis? So in chromothripsis, one chromosome shatters, but in chromoplexy, multiple chromosomes break. In chromothripsis, there are hundreds of fragments. In chromoplexy, there are fewer breaks. In chromothripsis, it's a massive catastrophe. In chromoplexy, interconnected rearrangements are there. In chromothripsis, it's like one book exploding. In chromoplexy, it's like several books getting mixed together. Which cancers show chromoplexy? Prostate cancer, sarcomas, leukemias, and some solid tumors. So chromoplexy in short is when several chromosomes break at the same time and their DNA pieces become incorrectly exchanged. I spoke about 4 phenomena recently, which are chromothripsis, Kataegis, whole genome doubling, and chromoplexy. Today I'll explain them simply and compare them. So what happens in each of these? In chromothripsis, one or few chromosomes shatter into many pieces. In Kataegis, hundreds of mutations happen in one small DNA region. In whole genome doubling, the entire genome duplicates. And in chromoplexy, several chromosomes break and exchange pieces. So an analogy or an example to explain each. In chromothripsis, one book explodes into hundreds pages. In Kataegis, ink is spilled on one of the pages. In whole genome doubling, every book gets copied twice. And in chromoplexy, pages from 5 books get mixed together. What is the effect on tumor suppressor genes? So, chromothripsis may delete tumor suppressor genes. Kataegis may mutate tumor suppressor genes. Whole genome doubling may make extra copies that would mask the loss. And chromoplexy may break the genes. Now, what is the effect on oncogenes? So, Chromothripsis may amplify oncogenes, Kataegis may mutate oncogenes, whole genome doubling makes extra copies that may be amplified, and Chromoplexy may create gene fusions. What is the result of all four of these? Chromothripsis results in sudden genomic catastrophe. Kataegis results in a mutation storm. Whole genome doubling makes evolution easier, and Chromoplexy results in a rearranged genome.